large amounts of fluid around the baby, sometimes even on the early scan. In the first trimester of pregnancy, there might be extra fluid behind the neck and the nuchal region. And as pregnancy progresses, sometimes that excess fluid around the baby, polyhydramnios, might be there. There might be a big baby, so what we call macrosonia. And there can be differences about heart structure that are seen antenatally or about heart rhythm. And again, I think Petra's going to talk more about this later. And when babies are born, really the sort of most dramatic thing for some of them will be this pattern of poor feeding, and sometimes low tone is part of that. And other differences might be there, and we can talk about those details where it's relevant later if you like. And as children get older, progressive short stature is something that will be an issue for many of them, and learning differences, again, are something that crop up. The way that we do genetic testing has changed in recent times. I'll talk a bit more about that later. There are an increasing number of people with these conditions who are picked up because they've had a broad-scale genetic test for a developmental set of differences and not necessarily had a diagnosis before the test result was known. So nearly always the gene change responsible for Costello or CFC syndrome has happened completely by chance, newly, in the sperm or egg that went to make the person. And that's just what de novo means. And obviously there is nothing that the parents have done or been exposed to that's predisposed to this. The only thing that we know of as a kind of risk factor, if you like, is that actually every time a cell divides, there's a chance for a misprint in that cell's blueprint, the genetic material, to happen. And that means that chance errors accumulate as cells divide, and so older parents are a little more likely to have children with one of these conditions, and it's particularly the dad's age, rather in contrast to chromosome differences, where maternal age is a thing that makes the chances different. Because sperm cells are dividing all the time, actually that makes a difference in terms of the chances. But when people look at testicular samples from chaps across the general population, they find Costello and CFC related gene changes in the testes at a very low level, and that's just emphasising what a very chance occurrence this is in the large proportion of instances. So here we've got a little graph of different conditions you might have heard of, reading around the conditions of the RAS-MAP kinase pathway. And nomenclature, the way we describe things, is really difficult, and I'm involved with Noonan and CFC guideline development, and actually the amount of discussion that's going on about, you know, what's in and what's out with these sorts of things. You know, are Noonan syndrome and Noonan-like syndrome with loose anagen hair similar enough to be considered the same thing, for example? Where does that distinction between Noonan syndrome and cardiofaciocutaneous syndrome happen? And I'll talk a little bit more about that later. There are some distinct entities, although even when we get into that, Costello syndrome, whilst we think of it as being a recognisable thing in its classical form, there are rarer variants in HRAS that don't necessarily correspond to a pattern of Costello syndrome. Legius syndrome I won't particularly talk about. It's generally a mild condition with brown patches, cafe au lait patches on the skin. And neurofibromatosis type 1, quite a common and important condition if you're a doctor to understand, because 1 in 2,000 people are born with it, but only some overlaps to what we're talking about today. So here we've got some pictures of people with CFC, and you'll recognise some of these people. In fact, some of them I think we might struggle to recognise them actually, given the passage of time and growth, but some of them do remain quite recognisable. And so you can spot your friends there, and obviously very many thanks to them and their families for the pictures. Let's think about genes and how they work. We've got 20,000 pairs of genes in the human genome, and they're packaged in these things called chromosomes, and we get one of each pair of chromosomes from each parent. And changes in genes cause differences to the proteins that affect how they work. So that's what we call the central dogma, and it's pretty old, but it works pretty well for the majority of explanations that we need to give for conditions like this. So the DNA is the storage mechanism for our body's genetic instructions. Those need to be processed via this set of molecules called RNA, and in most cases RNA makes proteins, and proteins are the building blocks of the cells and what actually make us up. So we've still got a lot to learn about how proteins work, because they're really complicated things and they don't work in isolation. So they're working together with multiple different proteins and other molecules, and kinases are a particular set of proteins that can act to control signalling through cells. And we can think of this in a number of different ways when we're describing how they work. It might be that they're playing tag, and so there's a sequential message that's passed down by a module being connected one to another, or like a domino effect. If you think of people who set up domino rallies, that those can branch, can't they, and they can cause all sorts of different and intricate patterns. Or we can think about that as parts of an engine, and it's at this point that I really feel anxious, because there will be lots of people in the audience who know much more about how engines work than I do. But anyway, the point is that individual proteins don't work alone. There are many other proteins that inhibit and chaperone them. And so what we've got here is not exactly an engine, but it's got some bits and bobs in it. And this is a diagram that's a simplification of how the RAS-MAP kinase pathway is thought to work. What it can't represent is actually the temporal, the time-related differences that there are, and also the fact that this pathway is interacting with multiple other pathways at any one time. But here you can see the known associations with neurofibromatosis type 1 being purple, spread 1 being pale purple, and then we've got Costello syndrome is special because HRAS seems to have a unique role. In terms of Noonan and CFC syndromes, there's more in the way of overlap, and you'll see that we've got here genes that have been associated with Noonan syndrome in the sort of paler turquoise, and then those more classically associated with CFC in the darker turquoise. And when we've got genes that are named similar things, we kind of think, well, actually, do they do interlocking things and similar things? And to some extent they do, but actually all genes are doing slightly different jobs to one another. There is a degree of what we call redundancy in terms of there being spare and backup functions for some of them. But the other thing to say is that this is when we're thinking about how the pathway is meant to work according to a standard blueprint. Once you've got a change in one of these modules, actually all bets are off. And so whilst we think about the ways of describing variants, as geneticists we think about loss of function, we think about gain of function, actually all of the time there's almost certainly more nuance to that. And particularly where we're talking about things that have got these very close interactions and trying to understand how that might be. So most of these conditions are characterised by a single spelling mistake in a gene, and a single amino acid being different can be enough to fundamentally change how the protein works. And because of that, these important changes cluster in particular key regions of the gene and therefore of the protein. And here from Martin Zenker's database, you can see some little schematics of the gene changes that have been found in HRAS and in BRAF. And here in HRAS you can see them fairly exquisitely clustered round about codon 12, with some at codon 13 as well, and just these very, very few other little bits that don't project very well. Now, these databases are as likely to have ascertainment differences as any other form of information that's out there, so there probably are other variants that haven't been as well represented if they're not as classically causative of Costello syndrome, for example. But still, that's a pretty impressive demonstration of how closely related the G12S gene change is to the pattern of developmental differences that we describe as Costello syndrome. For BRAF, that's the most common cause of CFC syndrome, the situation is a little bit more confusing, but you can still see here we've got this really nice big peak of people with this Q257R gene change, and that's something that most commonly, I'll say in a minute, is associated with a recognisable CFC pattern. You can then see there's some other things that are quite closely clustered with it, and then this whole sort of set of other gene changes The BRAF gene's a much bigger gene than the RAS genes collectively, so perhaps more chances for things to go wrong. But actually you can see that differences across a much broader chunk of it seem to be important in the pattern of people with CFC and Noonan syndromes. And so when we're thinking about this, we've got what we think of as one pathway, but there are lots of genes involved in many disorders. We've got common ones like NF1 and Noonan syndrome. We've got rare ones like Costello syndrome and CFC. And we've got many genes responsible for Noonan and CFC, and as you'll see, some overlap between those. So, thinking about Costello syndrome, G12S is the gene change that we find in about 80% of people with a recognisable pattern of Costello syndrome. And people with this will often have low blood sugars when they're born, they might have heart differences, and Petra's going to talk more about those, and the poor feeding. The other thing that is quite a recognisable clue, and in the days before there was a genetic test, was something that people had to rely on a lot, was the difference about the skin of the hands and feet and the characteristic hand posture that many of you who know people with Costello syndrome will be able to recognise, and those of us who are here today will also see in some of the people who are here. The learning differences are something that are apparent often from early in life, but ultimately can quite often be less severe as an older child and young adult than they appeared as a younger child. And the really important thing from a medical management point of view of Costello syndrome is thinking about that high tumour risk that really starts from very early on with embryonal tumours, including rhabdomyosarcoma, and that's something for which screening is recommended. CFC syndrome has got some overlapping features with both Costello and Noonan syndromes, and the effects on development are quite variable. There are some people who have severe learning difficulties and epilepsy, and actually epilepsy can be very difficult to treat in a number of people and have a knock-on on development in itself. The skin and hair differences are something that can be quite dramatic for people with CFC syndrome, but again, how people are as babies doesn't necessarily predict how they'll be as older children and adults. And the lymphatic differences, again, are something that affect a minority of people, but are something to be aware of in the bigger picture. Just a word about Noonan syndrome that we've known about for 50 years now. So Jackie Noonan was a cardiologist in the US who recognised in her clinic of congenital heart disease patients that there were this group of children who'd got short stature and pulmonary stenosis, and that combination was not particularly common. We kind of expect growth to be different in children who've got very severe congenital heart disease, but actually pulmonary stenosis of itself was not necessarily a big problem, was it, for lots of these kids? And she noticed that some of them had got this facial appearance in common, and that led the condition to be named after her. And so for people with Noonan syndrome, some feeding problems are very common, but often these will just be in the category of difficult feeding or needing a tube, a nasogastric tube for a few months, for example. Undescended testes are really common, and whilst learning differences are definitely part of the pattern of Noonan syndrome for many people, these are often very mild, and it's not rare at all for us to have parents who actually have Noonan syndrome who are only ascertained when they have a child who's got a more recognisable pattern born. There are some variants of Noonan syndrome that are associated with specific genetic differences, and this is an interesting one that we've known about for about the last 15 years, this gene called SHOC2, that it's one particular variant that's responsible for nearly all of the people with this pattern of differences, and what they often have that's not particularly well shown in this slide from the original paper is their hair doesn't grow, and so they often will not need a haircut until they're about 6 years of age. There are a variety of heart differences that people have. The feeding differences are more severe than we tend to see in Noonan syndrome, so lots of people with SHOC2 gene changes have needed gastrostomy feeding, for example, for feeding problems that have lasted well beyond the first year of life. And there are other overlaps that the more we look, the more we realise actually life is a bit more complicated than we might have thought about. And this is a slide that I've just borrowed from one of my more medically focused talks, where actually there are some things that have been reported in the Costello syndrome literature as being a really good sign for Costello syndrome, when actually perhaps what they might be is a really good sign towards one of these conditions of the RAS/MAP kinase pathway. And the nasal papillomas are a big part of the pattern for some people with Costello syndrome, but they are also reported in some people with SHOC2 variants, embryonal rhabdomyosarcoma, whilst Costello syndrome is undoubtedly associated with the highest risk, is something we've seen across other conditions of the pathway. And this neonatal atrial arrhythmia, that particular trace was found in a child who had RAF1-associated Noonan syndrome. So all of these are kind of overlaps, but what's good about that is that actually it helps us to tie together as a larger group of individuals with related differences. And again, this is something that Lisa has done enormous work with from the point of view of thinking about rasopathies as a large group of conditions that therefore do have more impact than something that's very, very rare. So this is just a very ancient slide from my thesis, thinking about at that time the number of patients we found with each individual gene change across the cohort of patients with Costello and CFC, and this does reflect a historical Costello syndrome cohort that had been meticulously assembled by my colleague, friend and mentor Bronwyn Kerr over many years, who once the gene was identified, these people could be tested retrospectively to have their clinical diagnosis of Costello syndrome confirmed. And so actually the frequency of Costello syndrome in that prospective sort of real life cohort is actually much lower than this picture would look at. But this group of people where we're thinking about CFC and severe Noonan sort of presentations is probably more representative. And so you can see there that peak corresponds to the Q257R gene change that's commonest in BRAF, this one to the Y130C change that's commonest in MAP2K1 or MEK1. A word about the way we describe things in nomenclature. I was saying a minute ago that geneticists shouldn't be allowed to name genes, and I probably still agree with myself, but we also have got numerous different ways of naming them as well, which is a bit confusing for normal people. So anything that's not clear, obviously, ask me or ask your own geneticist as and when you've got a question. This is just a picture of some of the hands and feet that you'll know well the features of, and we've already talked about most of those. And I think Mr. Stevenson might be talking about Achilles tendon things this afternoon, is that right?
David Johnson.
Johnson,
yeah.
Sorry. I'm not working out yet this morning. Heart disease, I'm not going to touch on in any detail because Petra's lined up here and we know she's come, so we'll definitely be hearing about that. But just thinking a bit about how common Costello syndrome is, that's a really frequent question that we get in these kind of fora. And it's actually quite a difficult question to answer, even though nearly everybody has got a recognisable gene change and nearly everybody has had a pattern of differences that's significant enough to mean that they've had genetic testing. But really this is a slightly historical cohort, but from a time when we had a really quite a good handle on what data was coming out, because we were one of only 2 places doing testing for Costello syndrome, and with the help from Colin and everybody in the support group, we did think we got quite good ascertainment of the number of people with Costello syndrome in the country. And this has led us to a birth prevalence of thinking about 1 in 400,000 or thereabouts. And that corresponds really pretty favourably with the Japanese birth incidence data that they've calculated more recently. And when we think about those people from our historical Costello syndrome cohort, we've got about half of them still living, and some of them who we've died, we know quite a bit about the information as to why that might have happened. And although it's very small numbers. And what you can see here is these are people who are kind of still doing fine. This chunk is the biggest chunk of those who died, and actually those were little ones who were terribly sick at birth. And whilst rhabdomyosarcoma is the biggest worry for many people thinking about the uncertainties of Costello syndrome, for children to get into the window of worrying about that, they've already done better than really quite a large proportion of those who are going to have the most severe pattern of Costello syndrome. So does the exact gene change in HRAS matter? G12S, accounting for nearly all of the variants, still has quite a lot of variability within that presentation, and we'll see that amongst people here today, because actually there are lots of chance things that are in the mix as well. Just because you've got Costello syndrome doesn't mean there aren't other bits and bobs that impact on how your development goes. There's a question about whether G12A, that's the second commonest variant, has a higher cancer risk, and I think that that's really not very well ascertained. There are people with this particular version of the gene who seem to have a high number of papillomas, so those little benign warty growths that happen particularly around the areas where skin meets mucous membrane, so around the nose, around the mouth, sometimes around the bottom and genitals. And I think the other thing to think about is, as I said before, that often people with very severe presentations may be lost early as babies. And so that, again, skews our understanding of the data and the overall picture. But there are different versions of the gene that definitely give a milder pattern of differences as well. So in terms of one of those is something called G13C, so that's in that little kind of just the little shoulder of the peak next to the G12 residue, and that does seem to give a more mild Noonan-like pattern than a classical Costello syndrome pattern. And at the other end of the spectrum is the little ones who've got G12V gene changes, and that's a gene change that causes the protein to work in a very different way. And that's why we actually see the G12V change that's here really quite commonly in samples of tissue that have been affected by cancer. So when that gene change happens in the context of the other gene changes that are going on in cancer, it seems to be much more common than G12S here. It's the classical Costello syndrome common variant. We realised we'd got a little group of patients in whom we'd found this G12V gene change, and actually nearly all of them have had a really very severe pattern of differences. And so those have been babies who probably haven't made it much beyond the neonatal intensive care unit. I think the longest that any of them have survived has been 18 months or so. Although there is one rare individual who's got this severe G12V gene change, but they've also got an extra add-on gene change that stops that version of the gene being expressed very much. And so that in itself has balanced out their pattern of differences, and they're not so severely affected. So how do we think about managing Costello syndrome? I don't know how I'm doing for time, not too badly. We think about making a confirmed diagnosis as being the key to that, really. And there are some people who have had a confident clinical diagnosis, and actually that's enabled genetic testing to happen in a very targeted way. I'll talk in a minute about how the progress of the genomic test directory doesn't necessarily foster that anymore. But thinking about feeding needs, heart needs, and the tummy screening for rhabdomyosarcoma is really important. Rhabdomyosarcomas can crop up anywhere, but it's sort of within the trunk and abdomen in particular that they're most likely to be cropping up and not necessarily readily detectable unless you do an ultrasound. A brain and spine MRI scan is definitely recommended, and again, that's something that Karen Grip's done a huge amount of work in the States about the progressive differences you can get with the base of the brain. Eye checks are important, and then really the sort of key day-to-day stuff is thinking about support group information. So our job as doctors is to make sure that people can be put in touch with the likes of you guys, and then there are management guidelines that have been drawn up in the last few years for Costello syndrome. And really, in terms of what's needed as an ongoing thing, it's almost a cut and paste slide, really. But as children grow up, orthopaedic complications are something that we'll hear more about this afternoon. Growth, again, is important, and Professor Clayton's going to speak to us about that shortly, and the neurological sequelae. So, just thinking about CFC syndrome, it's much, much commoner than Costello syndrome, and on my old version of this slide it said at least twice as common. I think actually we have at least 10 times as many new CFC diagnoses as we do Costello diagnoses. But because of the variability of this condition, it is a bit harder to know how it should be managed and what recommendations we should make. It's important to say there isn't any tumour screening that we recommend, but the risk of epilepsy can be high, and how we approach that again is largely by making sure that parents and primary caregivers are aware of the chance rather than by sort of doing anything extra. Again, the management guidelines, CFC International drew some up some years ago, and CFC is undoubtedly part of the Noonan syndrome spectrum guidelines that I'm working on with a group of about 70 collaborators across Europe and beyond, and that's to be finalised next year, and hopefully will be helpful for people, and we particularly want it to be helpful for people where there might be uncertainty at the moment as to whether it's Noonan syndrome or CFC syndrome, because actually whichever of those it is, wherever somebody's sitting on that spectrum, we want there to be guidance that might be appropriate for them. And so, does which genes altered in a person with CFC make a difference? It certainly can do. BRAF is much the most commonly identified gene, but the precise change within the gene might also be important as well. And so whilst for nearly 20 years now we've been talking to people about get your gene change known, just knowing which gene it is might not be enough in terms of understanding the pattern of differences. Here we've got some of my friends, I think all of whom I've met through this meeting in the first instance probably, who've got changes in the BRAF gene, and people will often have moderate to severe learning differences, although there are undoubtedly some people who have milder patterns of difference. Feeding problems are really very common, and I personally don't think I've met anybody with a BRAF gene change who's not had a gastrostomy because of the length of their feeding problems that have cropped up. The heart differences that people have are quite variable, and I've already said that significant epilepsy is a part of the pattern for a number of people with this condition. And what you can see across this group of young people who I think are all now teenagers or even older, is a large head with curly hair and skin differences. Thinking about other genes, MAP2K1 is a relatively less frequent cause of CFC syndrome, but there's one particular gene change in that that's got a real peak of incidence. And there's no prizes for guessing where I met this young man many years ago. Can't remember if it was this very hotel, I think it was probably in Northampton, that you had a Halloween party in 2010.
2010, yeah.
Some of these people with MAP2K1 gene changes have had severe epilepsy, but the other thing that seems more common in people with changes in this gene is contractures, so limitations to how their joints work. And these are quite uncommon in other people with CFC unless they're not very mobile at all from the start, if they've got very severe developmental differences. And there might be fewer heart and skin issues, and that's again where there's a slight problem with the way we describe things, because to talk to a family and say, I think your child has got CFC syndrome, when actually they have a normal heart, their facial appearances aren't remarkable, and their skin's absolutely fine, sort of is a little bit of a hard win, that. MAP2K2 gene changes are what appear to be a rare cause of CFC, and there are only small numbers of people known, but there are some big families, and you can see here that they've got quite a lot in the way of the sort of skin and hair changes, but much less in the way of learning differences. And these skin and hair changes can be something that evolve through time, and so dermatology input can be needed if people get multiple dark moles, for example. Those should be surveyed in much the same way that we would survey them in the general population. KRAS gene changes are also pretty uncommon, and that can be something associated with either Noonan syndrome or CFC syndrome. And again, I think that there is an important role in acknowledging the gene that's altered as part of the overall description of that person's pattern of differences. Because to say that they've got KRAS associated Noonan syndrome, say, well, perhaps they're at the milder end of the pattern of differences we associate with KRAS, or KRAS associated CFC syndrome towards the more severe end of differences. And there are a few people with specific changes in this gene who again have had kids of their own and passed down that pattern of differences that looks like more of a Noonan syndrome pattern. There are also people with KRAS who seem to have quite a high chance of having craniosynostosis, so changes in the skull bones. And this is something that's also been described in a few people with Costello syndrome, albeit quite recently, which is interesting given how closely scrutinised the group of people with Costello syndrome has been for really quite a long time now. So thinking about how we manage CFC, this is another cut and paste slide. You'll see pretty much from thinking about how we manage Costello syndrome, the feeding and heart differences are things that are the big deal for many young people with this. It's worth having a brain and spine MRI scan. And the chance of brain differences is that much higher in people with a severe pattern of developmental differences. And eye checks and support group information, again, is very similar. And the chance of orthopaedic differences is much smaller, except for that group of people with MAP2K1 gene changes we talked about before. So I'm all right for time, aren't I? I've got 5 minutes.
Yeah.
Think about how we do genetic testing. This has changed massively in the last decade or so. We've now got, whilst there is one gene to test for Costello syndrome, and we used to do a single test looking for that vast majority of changes within codons 12 and 13, we now have, if we're thinking about CFC as well, over 20 genes to think about for this group of conditions. And there's less than 10% of people where we think that this is the right clinical description where we don't find a definite genetic change after the best available testing. And that was on the basis of being able to do a panel test of about 20 genes up until a couple of years ago. But the wheels of progress have moved on, and the NHS Genomic Test Directory has been sort of overhauled, if you like, and so all the people who we see here today who've had a new diagnosis will actually have been tested through this mechanism where you can't order a test for Costello syndrome, you can't order a test for CFC syndrome, you can't even order a test for Noonan syndrome, which, given that we think up to 1 in 1,000 people might have it, is a little bit bonkers, but try telling NHSE that. So, in order to get our genetic testing for this group of conditions, we've now got lots of different ways of doing it, with different panels that I've listed there in terms of picking our genes. Most of the genes for these conditions are on most of these panels, and actually many of our patients will have at least one of these things that would be a reason why we could test them. And so in Petra's clinic, she'd been seeing people who might be eligible for the paediatric cardiomyopathy panel, for example. In Peter's clinic, he's got lots of patients with growth failure, and lots of people could have that test. There are then much broader tests in terms of differences with intellectual, developmental, or congenital anomalies, and the genes for Costello and CFC syndromes are on those panels. But you're actually getting all of the other genes tested as well as a bonus when you do that. And so that's what we'd call gene agnostic testing to a large extent. And that can be via a number of platforms I won't go into in detail. But the great thing about these gene changes where they're not there in mum and dad and they're newly in the child is that actually we've got fantastic control data for what's standard variation in that family. And so what we can do is a genetic test looking at mum and dad's genetic material and then just seeing what's unique to the child that's not been inherited from mum or dad. And this is actually a way of making a diagnosis of Costello or CFC syndrome really quite readily. And there is now testing that's available. There's a rapid test in a pregnancy where differences might have been identified, or where there's a baby or child who's very unwell, and that's with a 2-week turnaround. There's also a further category, which is where there's a child with developmental differences and the mum and dad are expecting again, and that's another situation in which a rapid test for the child with the differences can be thought about. And so that landscape's changing very much, but again, something that will be relevant to our cohort in the future. And the other thing to say is that the more we do this kind of testing, where we've not thought hard in advance exactly which genes to check, the more we find people with changes in the genes we're already interested in, who we didn't necessarily predict would have changes in those genes. So that again just keeps us on our toes in terms of understanding the breadth of differences that might be associated. Just a quick word about MEK inhibitors, and again I guess this might come up in the Q&A later, thinking about treatments. And so there are now MEK inhibitor treatments for people with neurofibromatosis type 1 that I mentioned briefly, and also for a very small number of people with Costello and CFC and Noonan syndromes, where these have been tried rather in an extremist kind of scenario where there's been terrible lymphatic differences or hypertrophic cardiomyopathy. And so that's something that is out there as a possibility, albeit not anything that's available for most people at the moment. And there are important questions, I think, for us as a community to address in terms of what outcome measures would be needed to be looked at, whether this is in the context of a formal clinical trial that might be really, really difficult to recruit to for very small numbers of people, or whether it's just as part of our kind of good clinical care and ongoing surveillance and understanding the effects of these sorts of agents in this group of patients. And so we could think about what the measures might be. Is it about getting home from hospital? Is it about how many other treatments people are needing to support them? Is it about mortality and the chances of actually surviving when actually they were in a very difficult situation to start with? And what would the appropriate longer term measures of the condition be? So in summary, I hope I've just sort of underlined to you that Costello and CFC syndrome might present in a very similar way, and that's why this conference is a lovely opportunity for families to come together. Costello has got specific risks, particularly of malignancy, and that CFC overall has got better survival than Costello syndrome, more variable developmental outcomes. And again, getting the right kind of input with community paediatrics and therapies can be really important for people predicted to have that condition. Just going to finish with a very incomplete but rather long list of thank yous. Any questions people would like to ask? Or you can just be brewing your questions for later when we've got the question session. It's absolutely fine.